Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on genetics. In the last lesson, we learned about the molecular structure of chromosomes. Today, we are going to learn about the structure of DNA and describe the 4 nucleotides. If you are prepared, we will begin by going over the basics. Deoxyribonucleic acid is one of the 2 types of nucleic acid. The other is, of course, ribonucleic acid, or RNA. This figure shows part of a DNA molecule. You can see that it is made of 2 strands joined together and wound into a double helix. If you look closely, you will see that the structures in the strand on the right are upside down when compared to the strand on the left. This is because the start of one strand is paired with the end of the other, making the 2 strands antiparallel. The basic unit of a DNA strand is a nucleotide. Students, can you remember the 4 types of nucleotides from your previous lesson? Take a moment to jot them down now.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello, students. I imagine that this was simple for you. The 4 nucleotides are as follows. Adenine is a purine derivative, which means that it derives from an aromatic organic compound. Cytosine is a pyrimidine derivative, also an aromatic organic compound that has a heterocyclic aromatic ring and 2 attached groups. Guanine, like adenine, is a purine derivative. It consists of a pyrimidine imidazole ring system with conjugated double bonds. Lastly, thymine is also known as 5-methyluracil. It is a pyrimidine nucleobase. Note that in RNA, there is no thymine-containing nucleotide. It is replaced by a nucleotide that contains uracil. All nucleotides contain three basic components: a phosphate group, a pentose sugar, which is deoxyribose in DNA nucleotides, and ribose in RNA nucleotides, one of the 4 nitrogenous bases. DNA is a huge molecule. The nucleotides are held together by bonds between the sugar in one nucleotide and the phosphate group in the next. This is sometimes called a sugar-phosphate backbone because it holds together the structure of the DNA molecule in much the same way as your spine holds up your body. The nucleotides in one strand are paired with nucleotides in the other, according to the base pairing rule, which states adenine-containing nucleotides will always be opposite thymine-containing nucleotides. Cytosine-containing nucleotides will always be opposite guanine-containing nucleotides. Because of this rule, all the bases on one strand of a DNA molecule are base paired by hydrogen bonds to those on the other strand. We say that the sequences of bases on the two strands are complementary. Another result of the base pairing rule is that the amount of adenine in a DNA molecule will always be the same as the amount of thymine. Similarly, the amount of cytosine will always be the same as the amount of guanine. If you know the percentage of just one base of a DNA molecule, it is possible to work out the percentage of all the others. Let us try to do this in an activity. Suppose that a given molecule of DNA contains 19% adenine. What percentages of the other 3 nucleotides must the molecule contain? Take a few minutes to solve this problem.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, students. You may find that the correct answer is deceptively simple. If the DNA molecule contains 19% adenine, then it must also contain 19% thymine. 19 plus 19 equals 38, which leaves 62% of the molecule unaccounted for. But we know that the remainder must be made of cytosine and guanine, and they must be present in equal quantities. 62 divided by 2 is 31, which is both the percentage of cytosine and of guanine in the DNA molecule. Good work. DNA is a very stable molecule at normal temperatures. The hydrogen bonds that hold the two strands together are individually very weak, but there are so many of them in the whole molecule that the two strands are held firmly in position. The bonds that hold the nucleotides together in each strand are much stronger than the hydrogen bonds. This is very important because the stability of the DNA molecule is critical in ensuring that the genetic coding which it contains does not become corrupted. We have time for one last activity, students. Here is a diagram of the basic structure of DNA. Please make a working copy of the diagram. Then, use what you have learned in this unit to label each element correctly. Do your best, and I will see you in a few minutes.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. Have you finished your diagrams? Let us see what the correct labels are so that you can compare them to your own work. This is the sugar phosphate backbone, and these are the base pairs. When we take a closer look, we can see the individual base pairs. Notice how the two strands are anti-parallel, running in opposite directions. This is an example of a nucleotide, and these are hydrogen bonds. Does your diagram look about the same as mine? If so, great job. Teacher, please check the student's diagram after the lesson. Today, we have learned about the structure of DNA and the 4 nucleotides. Until next lesson, thank you, teacher. Thank you, students.